For those who just joined, I'll just reintroduce myself. I'm Dilara Diken, and I just recently graduated from Bahçeşehir University. And I did my internship at Pine Biotech in the OmicsLogic Research Fellowship Program. And my project was on the threat that radiation poses to humans in space. And I analyzed the adverse effects of specifically high LET radiation on the mouse gut microbiome and how this affects the nervous system and causes psychological disorders. So I'll give a short introduction of how bioinformatics is involved, and then I'll give an introduction to my project, specifically how I analyzed my data, and then I'll be sharing my results and how I concluded this. And I think that bioinformatics is really important because during my time at the wet lab, I realized that we're producing such great amounts of data that we need to process, but the speed of processing that data is not fast enough. And bioinformatics and the machine learning and other new technologies that are involved are a great way to speed this process up. And another thing that I'm fascinated by in bioinformatics is that the same raw data that is collected for one project can also be used for other researches as well. And so I want to shortly talk about my experience at Pine Biotech and why I joined it. And it was because they have courses for everyone from beginner, and then they bring them up to a more professional level. And that's why I had an amazing experience, especially joined by my wet lab experience earlier. So it was like seeing the bigger picture. And to start with my project, basically it was on the prospects of space travel and how the advancement of space travel affects the astroscientists in space. And as Subrajit was talking about in his presentation, a lot of different fields are involved in astrobiology, and they don't necessarily have to be simply about looking for life in space, or, they don't have to be focused on one thing or the other. And that's why I decided to focus my project specifically on the health of astronauts and astroscientists. And as you can see here on the chart, on the graph on the right, the ISS, which involves different countries and different agencies, has started doing their projects in numerous different fields, including biology and physical science. And I decided to choose this, the health of astroscientists specifically, because to be able to continue this research out in space, we have to have healthy scientists out there. And I also think that the landing of Perseverance and the speed up of our space travel goals as humanity, especially with starting colonies out in space, it's really important to focus on research that affects the human health. So there are different types of radiation out in space, including ionizing and non-ionizing radiation. And my focus was specifically on ionizing radiation because it has high energy and this can cause chemical reactions. And I will frequently be talking about something known as high LET radiation, and this stands for linear energy transfer, which is a dosimetry. It's a measure of the effect radiation has on materials. And this is a very curious type of radiation because high LET radiation can easily be shielded for larger structures, but it can pass through microscopic structures such as cells. And as we know, radiation can have various effects, from major effects like cancer to minor effects like attention loss. And as you can see here, this was taken from a report from NASA, and they were analyzing how these listed issues will be controlled or uncontrolled in the next 10 years. This was a 10-year plan for the planetary mission. And as you can see at the bottom, radiation exposure to human health is greatly uncontrolled. Whereas above you can see other things like bone fracture or reduced aerobic capacity and host microorganism interactions. And these are partially controlled, or we're hoping to control them by 2028. But what we need to understand is that the radiation exposure affects all of the above disorders. So another reason why it's extremely important is because, as you can see on the right, the 6 months on ISS, on average, the radiation exposure is 10 times as much as the abdominal CT scan. So it is extremely important to consider radiation in our studies. So for my methodology, I separated it into 3 different steps, major steps, and this was data exploration, statistical analysis, and my conclusions. So for the data exploration, first, I found a data set from NCBI, which contained 80 samples from the mouse gut microbiome, and these 80 feces samples were separated into 2 categories, and basically this was based on radiation and days. So there was 0, 0.1, 0.25, and 1 GY radiation, and the samples were collected 10 and 30 days post-radiation. And then using the T Bioinfo platform, I used the Data2 pipeline and the PhyloSeq pipeline to find the abundances and also produce graphs including the PCA, NMDS, and Shannon index, which I will be talking about in detail. And to ensure that my observations from these outcomes were correct, I conducted some statistical analysis, including ANOVA and t-tests. And as for the drawing conclusions part, I was particularly focusing on lower doses since they penetrate cells more easily, and I wanted to see if the days also play a role because it's important in the acclimatization of bacteria. And also because these bacteria can be targeted for therapy for different diseases. Here we can see an NMDS graph, which stands for a non-metric multidimensional scaling graph. And here, as you can see, my focus was on days because I saw a better separation between 10 and 30 days post-radiation. And as you can see for yourself as well, the 10 is circles and 30 is triangles. And the 30 days post-radiation was more spread out throughout the graph. Here we see a Shannon index graph, and this involves the biodiversity of the samples, and it measures the richness and evenness of the species in the sample, which is equivalent to the effective number of species. So here, as you can see, when the comparison is made between all 4 doses of radiation, lower doses, which is 0.1 and 0.25 GY radiation, the biodiversity decreases in the samples. And then this was another graph output from the PhyloSeq pipeline. And for here, we can see several different things at once, including how the doses and days affect the abundance, and if these OTUs, which are operational taxonomic units, which will be equivalent to the species, as you will see later on, and how these are related to each other. And one thing that we can see is that OTU 4 and 5 are related, and we can also see that for compared to 0 GY radiation, 0.1 and 0.25 at 10 days shows lower abundances of bacteria. So I decided to focus on these, and I did a statistical analysis. First, I started out with ANOVA, which I used 3 doses, 0 GY for my control and 0.1 and 0.25 GY because from the previous phyloseq out pipelines, I told you, the lower doses were more significant. So I decided to focus on those. And then my outcome was that the days did not have as much of a significant effect, but the dosages did. And between these two variables, there was no interactions. So I conducted a t-test, keeping the days constant and just analyzing the doses of radiation compared to my control group. And I found that these four OTUs, OTU 3 and 4, which are related, and 5 and 8, which are related, as you can see in the heat map as well. These were the significant OTUs, and these were significantly lower at 0.1 GY radiation when compared to 0.25. And this was according to my threshold that I set at 0.25, 0.05 p-value. And I wanted to also check the abundances because it is important to see which families and genus of species increase and decrease, and if these correlate to my statistical analysis. Here you can see that I compared those 0 and 0.1 GY, and the Lactobacillus bifido erysipelotrichosis families decreased, whereas the Verrucum microbia increased. And when I conducted profiling and aligned these results with my t-test results, I found that OTU 3, 4, 5, and 8, as I mentioned previously, were in the Lactobacillus and bifidobacterium family. And as you can see, the four species, Lactobacillus gasseri and johnsoni, and Bifidobacterium longum and castoris, respectively, were the results of my profiling. So why did I specifically focus on these? Well, that's because these four species of bacteria have metabolites that affect the nervous system through the gut-brain axis. And they do this through the enterodendritic and vagus nerves, which play an important role in transferring the stimulus from our guts to our brains. And these four species were associated with the production of GABA, which stands for gamma-aminobutyric acid, which is the chief inhibitory neurotransmitter in the central nervous system. And GABA is extremely important because when it's produced in low levels, it causes depression and anxiety because of the lack of inhibition of the stimulus. So there's constant stimulus. And these bacteria have protein coding genes called glutaminase and glutamine synthase, as you can see in this GABAergic synapse. They are GS and GLS. And these are the same genes that are in the GABA pathway of humans. So we can assume that as the abundance of bacteria that have these protein coding genes decrease, the amount of GABA in our nervous system will also decrease. So we have two pathways that we can take from here. And that is we can either supply the body with exogenous GABA, or we can give the patients probiotics. But exogenous GABA in previous research did not give the desired effect. So probiotics is a more reasonable solution, a more effective solution in this case. So I can conclude this and gather up the findings of my research by saying that according to my initial hypothesis, lower doses of high LET radiation has a greater impact on the gut bifidobacterium and lactobacillus abundances, especially at 0.1 GY. And the acclimatization period can be deduced as 10 days. That's because at 30 days post-radiation, when we analyzed the samples, we did not see a significant difference. On the contrary, the abundance levels were slightly increasing. And this could be a result of the length of exposure, because we did not expose these mice to radiation for very long. And I think that this can be further research using combined effects of radiation and microgravity, and also increasing the length of exposure to see how it affects the nervous system and the abundances of bacteria. So we can easily say that this was a precursor study on how probiotics can be used as a treatment in psychological diseases and how radiation affects the nervous system. And sorry, yes. As for my limitations, one of my limitations was in taxonomic profiling because, as you know, in bacteria, there are sequences that are very similar, especially because for profiling, we use 16S regions of bacteria, and for the same genus, they are very similar. So it's difficult to pinpoint which species of bacteria you're dealing with. And another one was that the optimizing the parameters of the pipeline, but this helped me improve my approach to technical problems in coding and dealing with machine learning and its parameters. Also, another limitation that was caused due to the collection of samples in the wet lab was the sample size and doses. There were 10 samples per category, and if this sample size increased, we could have more accurate results. And we should also have in between doses, in between our 4 categories of doses. And we also look forward to research conducted on humans and astronauts directly. And that's because as much as we can analyze samples from mice, we do not know how the mechanism truly works in humans. So there are some researchers that collect samples from astronauts, but since the durations that astronauts are out in space is much longer, that is another limitation for those researchers. And here is my references. And I'd like to thank everyone for attending and listening. And I would like to thank everyone at Pine Biotech for their help. And especially Harpreet Kaur and Ilya Brodsky, because they were my mentors and they helped me a lot during this research. And thank you to Rutvi for initiating the Sciomics seminars because I think they're quite helpful.